It's going to make up our third and final video of cell respiration. This is for sections 2.8 and 8.2. And in this video, we're going to wrap up our discussion of cell respiration, specifically aerobic respiration, and talking about the final step in this process, the step that really produces all the ATP, and that is the electron transport chain. We'll also take a look at the structure of mitochondria and look at some of the different structures we've been seeing used in this different process. So the electron transport chain is the process of using electrons and concentration gradients in order to produce ATP. And this diagram here does a nice job of outlining what's happening. Within the matrix, we have an accumulation of NADH and FADH2 that was produced during the link reaction and the Krebs cycle. And those NADH and FADH2 molecules are going to donate or release electrons and hydrogen ions. And so the NADH becomes NAD+. The electrons are released to move through a series of enzymes in the inner membrane of the mitochondria. So this gray portion here is the inner membrane. Here the white portion is the matrix, and here is the inner membrane space. Electrons are released that move through these different enzymes in the inner membrane space, the inner membrane, and then hydrogens, so the hydrogen of the NADH, then moves through that membrane and collects in the inner membrane space. And as the electrons move through these different proteins within the inner membrane, it provides the necessary energy to move more hydrogens to accumulate them in this inner membrane space. The structure, as we'll see a little bit later, of the inner membrane space is actually pretty small. And the purpose of that is to allow a high concentration of hydrogen ions to build up very quickly. So lots of hydrogen ions build up in that space. And as we talked about earlier in the year, molecules move from areas of high concentration to low concentration. And so if there is a large accumulation or concentration of hydrogen ions in this area, they're going to naturally move to an area of low concentration, and they can do that by moving through this enzyme here called ATP synthase. And as the hydrogen ion moves through this enzyme, it almost acts as like a motor or a rotor, and it causes the ATP synthase to turn. And as it does so, that combines ADP, adenine diphosphate, with a phosphate molecule, a third phosphate molecule, to make ATP. And that is how the ATP is actually being created. The hydrogen ions then bind with used electrons. You can see right here they'll use electrons and oxygen to form water. And so for every 4 hydrogens that are used and every 2 oxygens forms 2 water molecules. And so this is that's very simply and basically what's happening to produce those ATP molecules. Oxidative phosphorylation is this process of ADP being phosphorylated to produce ATP. And that is through the oxidation of NAD+, mostly NAD+, a little bit of FADH. And through this process, a phosphate is being added to ADP. The energy that's being released by NADH and FADH2 is used to, these electrons that are being released, are used to move the hydrogen ions, as previously mentioned, through the electron carriers into this inner membrane space. And so as these electrons are moving through the proteins in the inner membrane, then that causes those hydrogen ions to move across the membrane. And so this is the process overall of the electron transport chain. We'll actually see this same process in photosynthesis in plants, but this is what is producing all of that ATP. And so the whole purpose of the steps previously was to produce NADH and FADH2 in order to provide a substance to power this overall mechanism. And so now let's take a look at the topic or process of oxidative phosphorylation. We've kind of already looked at this, but really what this is doing, a definition for this, is the accumulation of hydrogen ions moving across a concentration gradient. And as this is occurring, this movement is used to make ATP through ATP synthase. So the electron transport chain creates a high collection of hydrogen ions in the inner membrane space. We just talked about that. And that creates a concentration gradient. And the only way for those hydrogen ions to be able to move from an area of high concentration to low concentration is to do so through the ATP synthase, which then turns as the hydrogen ions move through it and couples ADP with a phosphate to make ATP. So again, this whole process is producing ATP. Oxidative phosphorylation by chemiosmosis is the synthesis of ATP from ADP. And so it's using energy of existing ion concentrations to power the ATP synthase enzyme. Hydrogen ions, again, moving from areas of high to low concentration. And what is really important here is that oxygen is the final electron acceptor. So those electrons that were used in the that moved through the proteins in the inner membrane that force the hydrogen ions to move across the membrane, those are accepted by the oxygen as well as the hydrogen to produce H2O. And so that's the last step of aerobic cellular respiration, and really where we're seeing the bulk production of ATP that cells need in order to be able to do different work. Let's take a closer look at the structure and function of mitochondria. It has some specific different shapes and different recognizable features that you can actually see in an electron microscope image. There's an outer and an inner membrane to the mitochondria, and this is actually a detail that helps to support the endosymbiotic theory of eukaryotic cells having an outer and an inner membrane. And the folds or projections are called cristae, and the space in between is called the matrix. And so this blue portion here is representing the matrix, and this is where the Krebs cycle and the link reaction are taking place. The cristae would be the membrane that contains the enzymes that are used in the electron transport chain, and then in between the inner and the outer membrane is the inner membrane space that we're discussing. So the matrix contains ribosomes and enzymes used during the Krebs cycle. The inner membrane holds electrons that are used in the chain in order to make ATP. The outer membrane separates the mitochondria from the rest of the cell and provides an environment that's conducive to respiration. The space, as previously mentioned, between the inner and outer membranes is really small. And so this small volume allows the hydrogen ions to build a high concentration very quickly, which then allow for that high to low movement of the hydrogen ions in order to power the ATP synthase enzyme. The cristae are tubular projections of the inner membrane, and really what these are doing is helping to increase the surface area so that oxidative phosphorylation can occur in more space and more area. That wraps up our discussion of respiration, both aerobic and anaerobic. We'll see parts of these same things, specifically the electron transport chain, in the next section of this unit looking at photosynthesis.